All right, so in part three, what they want you to do is something you've already done on the actual charts themselves, which is to try to identify which of the three hypotheses is the most parsimonious. And remember, the word parsimony or parsimonious actually just means the simplest, right? Which one has the least, which one follows Occam's razor the closest. And so when we're dealing with figuring out which one is the simplest, we look for the one that has the least number of evolutionary events necessary to reach the relationship demonstrated by the cladogram. So in number 2 here, you see that this particular set, there's 3 of them. There's moon eye fish, eels, and anchovies. And these 3 have this common ancestor here. The reason these are drawn is that there's no way of telling which one of these 3 is more closely related to one over the other. And so because there's not enough evidence to make that decision, they're put in this sort of grouping. There is evidence, according to this cladogram, that these 3 are more closely related than Gar. That Gar has a common ancestor that's further back. Some structure, some lack of common structure, or some structure that these have that this one doesn't, demonstrates evidence that these 3 are more closely related to each other than they are to Gar. So here they ask you to decide which of the 3 hypotheses, and this is really the big thing here, the big take-home, is that these cladograms are hypotheses, and they change as new evidence comes into light, whether it comes from the molecular evidence, from the fossil record, from comparative anatomy. The molecular evidence might be DNA, might be RNA, might be ribosomal RNA, might be proteins, and of course the comparative anatomy would be the structural differences between the two. It could be behavior, it could be where they live or what they eat. So in any case, all this evidence comes in to help develop this hypothesis, and the more evidence you have, the more sure you are that the hypothesis is probably correct, but we're never sure it's what's going on, because we're talking millions of years of time and relationships that are derived, or that are supposed from evidence that we've collected in the present time. However, when you look at the example of the whale that we looked at on the video that I sent you in Jupiter Grades, and we look at some of the other examples that we've studied in class, we're pretty sure that the relationships make a lot of sense under the umbrella of evolution, the way we've drawn them. All right, so let's look at part 4. Now here we use the word homoplasy, and homoplasy is actually being used as a replacement for this word that we've discussed, analogy, right? And so analogy is the difference between homology and analogy, when we're analogous structures versus homologous structures that we've discussed in class and you've been quizzed on. In this particular lab, they're using another word that means the same thing, homoplasy. And for something to be homoplasious is the same as to be analogous. And so in other words, something that's homoplasious, as you can see here in the writing, another thing that's homoplasious is due to convergent evolution. And convergent evolution, of course, we're talking about things that are not related via a recent common ancestor, that have similar traits, or traits that look like the traits that the others have. And the example that we used in class were butterfly wings, bird wings, and bat wings. and pterodactyl wings, and then all four of them had different most recent common ancestors, but they all developed wings in using different structures. For instance, a butterfly wing is completely different structure than a bat wing. Where homology, the word stays the same and means the same thing, which is that these structures, the pentadactyl structure of the various organisms we discussed, do indeed come from a most common recent ancestor. So they're asking you to choose character states that are shared by taxa to convergent evolution, and why are they shared? So again, they want you to look at this and figure out which character states are shared due to convergent evolution. And I'm assuming when they ask the question why, they want you to answer, why do you think that that is convergent evolution? All right. Okay, so here in this last question in part 5, they're asking you to take a look at the cladogram that you chose to be the most parsimonious. So that's probably the most accurate. And they're giving you this information. This is new data. And they're saying that all the other organisms on that cladogram have these number of openings, either 1 or 2, and peripheral are NA, not applicable. And they're asking you, well, how many do you think Annelida would have, given that if you placed all these on your cladogram, whichever one Annelida is most closely related to probably shares that same digestive system opening. Since they share so many other characteristics, it's likely they share this characteristic. So what they're asking you to do is this idea of inference, or taking information or data that you're given and knowledge that you have, and making a logical guess as to what you think this species, how many digestive system openings this species would have. There's no way of telling, really. I mean, they could have 3 or 4 if it had a mutation, obviously, that was different from its nearest relatives. But you can make an inference, you can make a logical guess, and you're probably going to be right. So look at the data, look at your most parsimonious cladogram, and put these traits on that cladogram, and then make an inference as to what do you think that an Annelida would have if you consider that its most common, what the organism that it shares the most number of traits with has. So, for instance, if Annelida's closest relative were Mollusca, then you might guess it has 2 openings. If it's Cinderia is its most common ancestor, you might say it has 1. In any case, take a look at it, make a decision, and put that on there, and we'll discuss it in class. All right, have a good one. So finally, we get to the last 4 questions in part 6. Okay, so for part 6, number 1, it's telling you that there's different categories of evidence that are in the matrix, the data that you have, the matrix, the facts. And there's particularly 2 specific kinds, and there's one which is the Hox gene, which is molecular. So there's this gene sequence that specific types of animals have that they call the Hox gene. It has to do with that a particular taxa have evolved from a common ancestor. This is molecular data. And then there's morphological data. Morphological data is the shapes and sizes of things like whether they have a pentadactyl anatomy or whatever the other characteristics in the matrix were. I think some of the things were radial cleavage and that type of morphological data. And in fact, gee, radial cleavage would actually be in another category than what we would say would be embryological or developmental, right? So developmental or embryological. So that would be for things like radial cleavage, or is it, or is it, or did it have bilateral symmetry and radial symmetry, or did it have an endoskeleton or exoskeleton and such? So most of these are morphological. There was one example, the cleavage, that we would call developmental or embryological. And then there's things like, exoskeleton, endoskeleton is, bilateral symmetry, etc. Those were morphological or structural, right, or structures. So these are things you can see and measure, are they symmetrical bilaterally or radially or what have you, where the Hox gene is molecular. So these two are similar in that you can see them, you can look at them under a microscope, or you can look at them with just your naked eye, and you can identify and classify. The Hox gene is something you can't see with your naked eye. You have to use biochemical processing, gene sequencing, in order to see it, and that's true for all molecular data. Now, what are the advantages and disadvantages of morphological versus molecular characters? Well, molecular characters like gene sequences and RNA sequences and protein sequences, these things are very accurate. They're very unique. They have they're very long sequences, and if two things match very well, then there's a high likelihood that they inherited this information from a common ancestor. So they're very unique. They're very accurate. They're very detailed. So there's a lot of information there. A single gene sequence might be a few hundred, few thousand nucleotides long, and yet they're going to be unique. They're going to be accurate. And it's very reproducible and can be found in different tissues, especially with genes. So that's the positive side of it, the molecular data. The negative side of the molecular data is it doesn't last long, right? Because if you think about it, bones can fossilize, DNA can't. So it doesn't last long. I think the half-life of DNA is 10,000 years. That sounds good, like a long time, but not when you're talking about fossils, right? So, and that's in perfect condition. In the real world, bleach and such will get rid of the DNA and the RNA. It'll break it down. You have bad conditions, and in bad conditions, we're talking hours, not thousands of years. So it doesn't last. So DNA evidence is an issue because of the time. The other problem with DNA evidence is you have to have unique skills to get the information. You have to be very well trained, and you have to have equipment, right? You're not going to be able to just look at something and tell the DNA characteristics. So these are kind of the negatives of DNA or molecular data, and these are the positives, right? So these are the advantages, and these are the disadvantages, right? So I think those are obvious. I hope that they're obvious to you. Morphological data is kind of, again, I think the advantages are that they last a long time. So, some morphological data on fossils have lasted millions of years. The other, some other interesting advantages of morphological data is you can see it with the naked eye. might use a ruler or a book to compare things to, but your eye is all you need to do to see it. So another advantage is there's very little training. With very little training, you can have someone identify specific structures, and by very little training, I mean months of training, because this is still a skill. But the training that it won't require years of specific training on how to sequence DNA. So little training, you just see it with your naked eye, it lasts a long time. Some of the negatives are that morphological data can be subjective. If not done right, and even when done right, it can still be subjective. So you say it's round, I say it's not as round. So some of the characteristics can lend themselves to be, well, in the gray area. All right. The other thing is that they're not necessarily very specific, right? They're not as accurate. Although you can make measurements and you can say, we can start talking about the ratio of the distance between the eye socket and the nose socket and things like that. There's some things that we can make more specific. They're nowhere near as specific or as unique or as accurate as genetic information or molecular data. So there's some advantages and disadvantages of both molecular and to morphological data. So both molecular and morphological data have advantages and disadvantages. You know, one really great example of some morphological data is very ambiguous, very not specific, very subjective, is this idea of elongate cylindrical body. And we even got to some of these discussions in class. So why is it that this creature why do we call it elongate cylindrical body when it's flattened? Well, do we call it elongated cylindrical? Do we call it flattened? And is that a different trait? Should we put it in a different category? So these things that start to become gray, start to step into the gray area of life that's not exactly black and white, and we're not sure. You know, I think one of the species had bilateral symmetry when it was in the early part of its life, and radial symmetry at the later part of its life. Well, which do you call it? You know, we have that ambiguity of molting. When do we call molting? Do we call molting only when it's molting for growth, or do we call molting when anytime you shed any kind of outer skin or epithelial layer? So there's that ambiguity. There's this subjectivity. There's those discussions that we go back and forth. Those are missing in the molecular world because it's very specific. It's very almost mathematical. In fact, it is mathematical when we're talking about whether something is right or wrong. So you get a much more black and white view of the world when you're talking about genes. Now, another problem with genes is just because that's the gene sequence, it doesn't mean that that's the structure that's going to pop out. So you have this Hox gene that does this thing. This is what it does normally. How do you know that that's what it does morphologically in a creature that was 10, 20 million years old, 10, 20 million years ago? So if, for instance, a gene produces a structure in an organism now, are we sure that that particular gene, we'd expect that gene to be found in an organism that's 20 million years old when that structure is present, or could another gene have made the same structure? And so could this be an example of convergent evolution where two different genes are producing the same structure? So there's some difficulty with that as well when you're working backwards. But overall, your molecular data is going to be a lot more accurate and a lot more dependable than some of the subjective traits that we look at. Other subjective traits are pretty clear, and you've seen that in this lab. Okay, something that's come up over and over again is this idea of most recent common ancestry versus just common ancestry. And the implications is really, really straightforward. Everything on the planet that's alive has common ancestry. We all have ancestry that can reach back to the first creatures, the first creature that was that came into being. So that is a common. We all have common ancestry. If we go far enough back, we'll find similarity between the bananas and humans. We'll find similarity between, insects and humans, etc. And that similarity means something. It means we have a common ancestry, and there's things that that there are implications to that. But the most common ancestor, the more recent ancestor, is much more useful because it tells us the more recently we're related, the more traits we have in similarity. As the activity that asked you to look at, digestive system openings in the previous slide, you can make inferences based on the data that you have, and make decisions on what you think this creature might be, or what kind of characteristics this creature might have, considering that it's in this group. You might be wrong, but you probably have a good chance of being correct, because being part of a group does mean something. There are characters that you share with these other organisms, and those shared commonly derived traits are not easily sloughed off. They requires an evolutionary event for that to happen. So question 3 asks you, how would you know if a character is a synaptomorphy? And the next part of the question is, what simple comparative method is used to determine this? So remember, a synaptomorphy is a commonly derived trait that's commonly derived among members of a taxa that share most recent common ancestor. So primates all have thumbs, for instance. What would be another example? Reptiles all lay eggs, for instance, or reptiles have eggs. Some reptiles actually hatch. The eggs hatch inside the reptile. But in any case, the point is that these are derived traits that come from a most recent common ancestor. And how do you tell if it's a synaptomorphy? Well, the easiest way to do that is to compare it, is to make your matrix, draw your cladogram, and compare the derived traits, and see if the entire clade has these traits. If the clade has these traits, then it's a synaptomorphy. Of course, if the clade has to have it, and it's more, then the outgroup must not have it. So if the clade has it and the outgroup doesn't, then you know that it's synaptomorphy. And could you involve fossils? For sure. You could take a look at a character and look at a fossil of, let's say we're looking at reptiles again, and we're looking at scales instead of live egg birth, or we go ahead and look at egg birth. If we find a fossil of a reptile that has a nest with eggs in it, the eggs can also fossilize, then we're pretty sure that organism that lived millions of years ago had that trait, and therefore it's a trait that was inherited by the whole group from that ancestor. So, and if we look at the whale and those hind legs, and we look at the present whale of having the pentadactyl morphology of being pentadactyl, we can look at the common whales today, and we can see the remnants in the back legs. We see the front flippers. They have the pentadactyl structure, and then we can look at their ancestors, look at the fossil evidence, and see pentadactyl structure in all 4 limbs, going back to that common ancestor between whales and hippopotami. So the bottom line is, the hippopotamus and whales share that common ancestor. We can look at the fossil evidence and identify pentadactyl structure as a definite synaptomorphy, or a commonly derived character from a most recent common ancestor. All right. All right, the last question is this idea of epidemiologists and phylogeny. If you haven't looked up what epidemiologist is yet, an epidemiologist looks into how diseases spread. How are they spreading? And so we're looking at the Zika virus recently, or we're looking at Ebola. An epidemiologist is the guy that sits there and starts to collect, a doctor usually, an MD, could be a PhD. And they're out there collecting the data from all the doctors that are reporting in illnesses, and they're collecting viral samples to study them to make sure that they are the disease that they think it is. And once they identify clusters, they can move backwards and find the patient zero and find out where it started, how it spreads, and how we can best stop the disease. So that's what epidemiologists do. Now, to use a phylogeny, how would the epidemiologist use a phylogeny? Well, if an epidemiologist is looking at, you can create a phylogeny, first of all, for a group of humans, right? We can look at a phylogeny that looks at genes. Especially if we look at the molecular data, we have to look at molecular data because fossil evidence won't be any good to us for humans, because we're all humans, and so we all share the vast majority of our characters. But if we look at our genes, there are many different gene sequences and RNA and DNA and mitochondrial DNA evidence that we can organize into more recent groups, and these are even more recent common ancestry, so we can find out where groups of people came from. And the key is an epidemiologist would look at that data so that they can figure out who are the most vulnerable of our population to this virus or bacterial infection, and decide how to best treat the humans for this disease that do get it, but also how to prevent humans that haven't gotten it yet from getting it. And so by using phylogenies, we can see, let's say, everyone with this particular gene sequence, or everyone from this region of the country is getting the disease. These are the most vulnerable people, the people with this with this gene or with this set of genes. We need to make sure we identify those people and keep them away from the diseased individuals. And these people are resistant to the gene, so maybe we can find a treatment for the disease using this group of people's natural resistance. So this is one way you can use a phylogeny to combat emergent diseases that are just coming out, that we don't have treatment for, that we don't know who's vulnerable to it, that we don't know where it came from, we don't know how it spreads. We can use phylogenies to look at what those most vulnerable groups are, and how does this virus or bacterial spread? Is it airborne? Is it through the water? What have you? All right, I hope this helped. Have a good one.